Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video, we'll be discussing another DABT topic, ICH-6 guideline. So we'll go, unlike other videos where we have taken questions from ABT recertification, here the question papers whichever I am having, no, the previous year's question papers in which in that no, most of the questions on ICH guidelines were not asked. But if you see recent 2024, 25, 23, 22, repeatedly these areas they are covering. So, if anybody who have appeared for the exam in 2024, 23, 22, if you have any questions related to, if you remember that some questions are with respect to ICH safety guidelines, please you can comment so that we can make or discuss about those questions. Um, but since we do not have any questions, I have taken the questions from Google AI. So we'll discuss this guideline in two parts. Another thing we have to remember with respect to ICH 6 R1 guideline is first guideline it came in 1996. Then in 2011 they added as an addendum. Okay, some of the things which are still need to be, because always understand these guidelines are dynamic. New information keeps coming. So based on new information, challenges which the industry face, pharmaceutical industries and scientists face based on that, these guidelines will be updated either in the form of addendum or appendix or in the form of revision. So this is S6R1 revision 1. Okay, so let's directly dive into the question. The question is, what is the primary objective of ISH S6R1? So whenever you are reading any of the safety guidelines, whether it is S1, S6, S9, or even including quality guidelines or multidisciplinary guidelines also, you need to know what is the scope, what is the objective. This guideline is applicable to what type of pharmaceuticals, what type of chemicals, and this guideline is not applicable to what type of pharmaceutical or chemical. So we'll quickly go through. So primary objective of this S6R1 is, the answer will be in the green text, that is to provide a framework for preclinical safety evaluation of biotechnology-derived pharmaceuticals. So it is not discussing about clinical trials, it is discussing about what are the toxicology studies, what are the preclinical safety evaluation need to be performed for this biotechnology derived pharmaceuticals. For small molecules, you have different guidelines. You have like you have S4 for chronic toxicity studies, you have M3 guideline is also there. But for biotechnology, large molecules which having a high molecular weight, complex molecules, proteins, biologics will be there. No, for them, ISHS1 R1 is the guideline. If you look into the name of the guideline also, you will understand. So what is the explanation? We'll see. ISHS6 R1 provides a scientific based framework. Okay, it is not a checklist. Okay, based on case by case, based on what is the nature of the biotechnology product, based on that you will be. That's why it is science based framework for the non-clinical, preclinical safety assessment of the biotechnology derived products such as monoclonal antibodies. So what comes under this bigger umbrella called biologics or biotechnology derived products is monoclonal antibodies, recombinant proteins, cytokines, and many more are there. Okay, that we'll discuss from the guideline only. Also see why the other options are not correct. First option was to establish GMP requirements. So GMP is something related to manufacturing. So you have separate guideline for GMP only. Then we have to define clinical trial endpoints. For clinical trials also you have separate series of guidelines to regulate marketing authorization. We are still not discussing about marketing authorization. We are not discussing about clinical trials. This is still very at the initial stage, that is preclinical stage, non-clinical stage. Pause the video. I would request you go back and also see, since here the term is given here, I would request you to go and check what is the difference between non-clinical and preclinical. Why certain times toxicology use the term preclinical, certain times non-clinical. In some other video, I'll explain you the answer. Okay, coming back to the guideline. So section 1.2 discussed about the objectives. So this particular guideline, which is in harmonization with three ICH countries, that is Europe, Japan, as well as US. And if you look this one, no, very important thing. All regions have adopted a flexible. As I told you, this guideline is not it's not concrete. It has some places where you can it has given you some areas where you can, based on your drug, biotechnology drug, you can fit in your fit in your drug into this. That's why it is flexible, case by case, like decision tree. It is not and science based approach. to preclinical safety evaluation needed to support clinical development and marketing or authorization. So this is both useful for clinical development as well as marketing authorization at the later stages. Don't get confused here. It is given to regulate marketing authorization, so don't get confused. In this rapidly evolving scientific area, there is a need for common understanding of continuous. Yes. So all these three global regions, that is important. So we will just quickly go through what are the primary goals of this preclinical safety evaluation is to identify an initial safe dose and subsequent dose escalation schemes in the case of human. So for this biotechnology-derived products, how to decide the first in human dose? Before initiate for the phase one initiation, what will the starting dose that you can get it from this type of guideline. This will guide you the next to identify the potential target organs for toxicity. So similarly, like conventional molecules, small molecules, you have repeat dose toxicology studies where you will be having a novel based on which you will be also have you will identify. Then primary objective of repeat dose toxicology is to identify target organ. So similarly, this ICH-S6 for biotechnology products, it will give you about a target organ toxicity and for the study of whether such toxicity is reversible. So the main purpose of having a repeat dose toxicology study with the recovery group is to identify if once the cessation of the treatment is there, is the whatever the adverse effects which we have observed, are they reversible or irreversible. Okay. And third objective is to identify safety parameters for clinical monitoring. So from these trials, no, so if we understand there is a potential concern that this particular drug is going to affect the heart or about the kidney or about the liver, so those parameters, those biomarkers, when you do phase 1 trial or phase 2 trial, you can monitor. So it will be a great help to the physicians to decide what type of markers and what type of adverse effects they should be monitoring in the phase 1 or phase 2 trials. So adherence to principles presented in the document is intended to improve the quality and the consistency of preclinical safety data. That's why you have to stick to GLP, all those things. So this was the thing. You can do one thing. You can download this particular guideline, ICH-6R1 guideline. You can take a print. You can compare and you try to read it. Okay. So you can pause and see also what are the different examples for biotechnology-derived products, biologics. Okay. Now we'll enter into question number 2. That is ICH-S6R1 mainly applies to pharmaceuticals produced from. So, how these are synthesized, how they are produced. So, the right answer will be in the green text, that is characterized cells using expression system. So, this is a method through which biotechnology biologics are produced. Chemical, other options will see chemically synthesis only, that comes for small molecules. Herbal extraction for herbal drugs, there will be separate guideline, and radioactive materials also separate guideline. So, you go back and check how you are going to what do you say monitor, or how you are going to conduct radioactive materials, what are the guidelines are there, you see. Okay. So as I told you, you have to read whenever it comes to safety guidelines, you have to read objective as well as you have to read scope. If you look into the scope now, it applies to products which are derived from characterized cells through use of variety of expression system. This is what the you should remember the guideline, you will be able to answer in the exam. Okay. Including bacteria, yeast, insect, plant, and mammalian cells. So these are the various from which cells you have to characterize those cells, and from that you have to extract it, expression. The intended indications may include in vivo diagnostic, therapeutic, or prophylactic use. The final use of biologics might be for various indications, for diagnostic, for therapeutic, as well as prophylactic use. Okay. So active substance, if you see, they are proteins and peptides, derivatives, and products of which they are components. They could be derived from cell cultures or produced using recombinant DNA technology, that is hybridoma technology, monoclonal antibodies, including production by transgenic plants and animals. Examples include, so what are those examples? Cytokines, plasminogen activators, recombinant plasma factors, growth factors, fusion proteins, enzymes, receptors, hormones, monoclonal antibody. Simple, I'll tell you how they will ask you in the exam. The question is that they will ask you, there are certain X company is there and they are preparing certain X Y biologic they are preparing. Okay. And they will ask you like question, what are the toxicology studies or what are the pharmacology, what PK studies or TK, or which guideline you have to follow. So you have, for that first you should know what is the nature of the chemical. If my nature of the chemical is a oligomer or aptamer or a monoclonal antibody or a hormone, then based on that you have to decide whether I have to follow M3 guideline, whether I have to follow S9 guideline, whether I have to follow S6 guideline. Okay. Then next, the principles outlined in the guidance may also be applicable to recombinant DNA protein vaccines. So, in addition, this list is exhaustive. In addition to that, recombinant protein vaccines, chemically synthesized peptides. So, peptides also comes under this one. Plasma-derived products, endogenous proteins extracted from human tissue, and oligonucleotide drugs. For oligonucleotide, we have separate. I think S3 or S13 or S12 guideline is there. We'll discuss in some other video also. So this is how you have to remember. So I would also request you. So if you're working in the biotechnology industry, so whenever you're working with certain biologic, try to understand what is the nature and see what are the how whether it falls and which is an example. Look for the examples. I'm saying plant derived are there, plasma cytokines are there. So look for the examples. Make a list, make a note. For enzymes, what is for receptors, what is for hormones, what are the examples which are already available in the market. Okay, so in addition to which this particular guideline is applicable, you should also remember that this guideline is not applicable for what? Because those not applicable only they'll try to give you in the exam as another options. So we have the document does not cover antibiotics. So antibiotics we have separate guideline. Allergenic extracts will not be. Heparin, vitamins also separate, and cellular blood component like serum or something like that, and conventional bacterial and viral vaccines. So they will try to confuse you. You will be thinking vaccines will come under this. Conventional will not come. Biotechnology-derived vaccines, recombinant DNA technology vaccines are there. No, those will come. Recombinant DNA protein vaccines. But the conventional bacterial and viral vaccines, they don't fall under ICH-S6. Okay, DNA vaccines or cellular and gene therapies. So gene therapies are also not under this S6 guideline. It is bit confusing, but still you have to try to remember. Guideline covers. So if you see the answer is here: proteins, peptides, here yeast, insect, plant, and mammalian cells. Okay. So we have covered objective. We have tried to cover scope. Now one question with respect to cell. You'll keep getting this word, that is species selection or selective species. So the question is, according to ICH S6R1 guideline, the most important factor in selecting animal species. So in the case of biologics, no, the biologic to go and bind, there should be a receptor, and that receptor availability should be there in the animal model in which you are conducting the test. Okay, so in case that particular animal, for example rat or rabbit or monkey, in that the particular receptor epitope is not there, then even if you give this biologic, it will not go and bind because there is no absence of receptor. So such kind of studies are going to give you no meaningful data. In fact, something adverse effects in case if you find, it will unnecessarily create a confusion and it will cause interpretation of the whole the particular biologic will, it will create a chaos. So in such a case, always we have to look how we are going to identify the selection of the species. You do some pharmacological from receptor binding studies you will be doing. So let's see what the guideline is saying. The right answer is pharmacological relevance. So most important factor whenever we are selecting an animal species is pharmacological relevance. Other options are cost, that is lower cost, largest body weight, ease of handling. So all these things, three things are not applicable. So the chosen species should express the target epitope receptor. If you see this one antibody, on this you have antigen, you have epitope. This epitope should be there or receptor should be there, and they demonstrate relevant pharmacological activity comparable to humans. So let's see what is the section 3.3 in the guideline says. Animal selection, animal species, and model selection. The biological activity, together with the species and of tissue specificity of many biotechnology-derived pharmaceuticals, often preclude standard toxicity testing design in commonly used species. Like we should not think our mindset should not be like that of conventional molecules. So the whatever the checklist standard battery of toxicology test for the conventional small molecules are there, no, we should not think in that aspect. Safety evaluation program should include use of relevant species. So relevant species is presence of that receptor, that you have to remember. That is a take home thing you have to remember. Relevant species is one in which the test material is pharmacologically active due to expression of that particular receptor and an epitope. In case of monoclonal antibodies, here you can see the figure. A variety of techniques, that is chemical and functional text can be used to identify relevant species. So this comes in the very beginning of the drug discovery phase only. So knowledge of this receptor epitope distribution. Once you identify the receptors, distribution of the receptors, how many are there, that also important. So knowledge of receptor epitope distribution can provide greater understanding of this potential in vivo toxicity. Okay, pause here. Once again, you read it or you go back to the guideline and read it. Okay, so third was with respect to selective species, that is relevant species. Now we'll discuss another question with respect to safety pharmacology. So we'll be discussing section 4.1. The question is, which statement about safety pharmacology in ISH S6R1 is true? So in case of S6R1, in case of small molecules or conventional molecules, we'll be doing FOB, that is functional observation battery. Safety pharmacology will be doing to understand these three organs, that is CVS, CNS, as well as respiratory system. Okay, so the right answer will be in the gene test. It may be incorporated into the toxicity studies when appropriate. So what this ISH S6R1 guideline or the addendum part, what it says that whatever the first came in 97, now it is in 2011. So always these guidelines to try to incorporate as much as possible 3R principle. So main one of the pillar of 3R principle is reduce, reduction of as many animals as possible, so that we can use less number of animals and get the same information. So what the guidelines suggest or the toxicologists or regulatory agency are saying that you do safety pharmacology whenever you are doing general pharmacology, that is repeat dose toxicology study. In that only, during that study only, during that phase of the study only, to the with that animals only, you also add another parameter, that is safety pharmacology, that is FOB or CNS related parameters or heart related parameters like HER, GSA, like that. Okay, you have to add to the repeat dose toxicology only. Same concept you will be seeing in the case of few OECD guidelines are there. Combined repeat dose reproductive and developmental toxicology, or certain times combined repeat dose extended carcinogenicity studies will be there. And nowadays you also have repeat dose toxicology in which micronucleus is also added with the same animals, like that. So let's see what the guideline is says. For many biotechnology derived products, safety pharmacology endpoints can be integrated into general toxicology study if scientifically justified. So we have to also give rationally also why we are adding one more parameter to this particular same repeat dose toxicology study. Okay, so safety pharmacology, aim of safety pharmacology study should be reveal any functional effects on the major. This is we already know. So see, this is the important point you have to remember. Where necessary to incorporate a particular monitoring for these activities in the toxicity studies and on the clinical studies. Okay, this is the thing you have to remember with respect to potential or undesirable pharmacological activity in appropriate models where necessary to include to the general repeat dose toxicology studies only. Pause here, try to read it. You also read safety pharmacology in the case of addendum section also in the guideline. Okay, now another question is with respect to immunogenicity. So question is, immunogenicity in animal studies primarily affects. So what is happening in the case of biotechnology is that if that biologics is going to trigger your immune system, then entire interpretation of this particular toxicology data will be changed. So how to avoid, how to address, how to overcome is that you have to do immunogenicity test for this biologics. So how we are going to do? We have section 3.6 is there. Whatever this, for example, we have anti-drug antibodies. They will may alter exposure pharmacodynamics. Okay, and other and observed toxicity, making toxicology data interpretation more complex. It becomes more challenging for the toxicologist if the biologic by itself is triggering your immune system. So that is the reason we need to immune related biomarkers or testing also we should be including in the protocol. So we have um, biotechnology derived pharmaceuticals intended for human are immunogenic in animals. Okay, therefore measurement of these antibodies associated with administration of these type of products should be performed when conducting repeat dose studies in order to help in interpretation of these studies. So this is the answer. Okay, so pause here and also try to read. See, interpretation of data is the main thing. it is going to. So what kind of you are doing is you are doing immunohistochemistry. Some biomarkers will be there. This titer, number of responding animals, neutralizing antibody responses should be characterized. Titer, number of responding animals, neutralizing or not neutralizing, and their appearance should be correlated with any pharmacological, toxicological changes. Okay. So next we will see another question with respect to, that is, tissue cross-reactivity. So something in extension to immunogenicity only. So question is, for monoclonal antibodies, a key requirement for species selection is how one is how we are going to, one of the key requirement for this species selection is tissue cross-reactivity. So in for biologics, tissue cross-reactivity is also one of the things what regulatory agency asks. So what we'll see, what is this one? So target cross-reactivity with human tissues is the right answer. So other things are not applicable. The other options A, B, and D. So the selected species should demonstrate relevant binding and tissue cross-reactivity to the therapeutic target. So what are TCR? Will see tissue cross-reactivity studies are in vitro tissue binding assay employing immunohistochemistry. This is IHC conducted to characterize the binding of these monoclonal antibodies and related antibodies like products to antigenic determinant in the case of tissues. Other technology can be employed in place of ICH techniques to demonstrate target binding site distribution. So something related to relevant species only, this particular section. This is given under note 1. Okay. So like that, we'll discuss another video, some of the rest of the sections of this particular guideline. So generally what the examiner asks, what are the exam key is, they will, today we try to cover, see, most frequently tested concept in ISHS 6 is they will ask you about relevant species selection, single species that we will be seeing, immunogenicity now we have covered, okay, and other points we'll discuss in another video. In the meanwhile, you can read. So in addition to that, I will also request you try to see, coming to biologics, we are discussing biologics here, also compared with biosimilars. So what are the additional, if any needed, any toxicology studies are needed? So difference between biologics as well as biosimilars, what kind of testing are needed? So like this also you can read. Okay, thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. All the best for your exam.